Good afternoon, everyone. My name is Thanawat. Today, I will present the lab project of silencing of SESN1 in the context of transcription 69 DEK NUP214 acute myeloid leukemia. My supervisors are Dr. Anna and Professor Jude. The ELN risk classification divides AML into three categories by using chromosomal and genetic abnormalities. One of the adverse or poor risk AML is transcription 69 or DEK NUP214 fusion genes that represent in about 1% of all AML. From the preliminary data of my host group on 50 primary samples of poor risk AML, the RNA-seq showed the homogeneous gene expression signature on the 4 cases of transcription 69, which different from the other abnormalities. The gene expression of transcription 69 also showed a significant increase of HOX gene and other 6 genes that compare to other poor risk AML. Moreover, the AML cell line study on gene expression also revealed that the striking increased expression of these 5 genes in FKH1 cell, which carrying the transcription 69 AML cell line, and only these 3 genes in OCI AMLT cell line. This study is aimed to decipher DEK NUP214 oncogenic mechanisms and identify therapeutic vulnerability. In this project, we select one of overexpressed genes in transcription 69 AML named SESN1 and create the model to silence this gene by using lentiviral plasmid that coding small hairpin RNA or shRNA. And then the SESN1 expression in the AML cell line that we shoot OCI AMLT cells were used to show the effect of shRNA silencing. There were 4 methods to create the model of silencing SESN1 by the shRNA. First is to produce the shRNA plasmid and lentiviral. Second is concentration and titration of lentivirus. Third is to transduce of OCI AML3 and then perform SESN1 expression. For shRNA plasmid and lentiviral production, the transfection of HEK290 T cell with the transfection mixture was prepared by using plasmid as transfer vector with gene fluorescent protein or GFP. The SH1 and SH2 that targeting SESN1 as well as camber shRNA that as a negative control were used, and the packaging vector as well as enveloped vector were also prepared, isolated, and measured the concentration by nanodrop. We choose calcium chloride as the chemically treated in transfection process, and then mix with Hep to stabilize the pH. All the reaction mixture were transferred to previously seeded HEK290 T cells with 20.5 million cells per digit and 60% confluency. After overnight incubation, GFP positivity was confirmed by fluorescent microscopy. To concentration the virus. After producing virus in the media that treated HEK cell for 24 to 48 hours, the media was collected, centrifuged, filtered, and underwent anti-centrifugation as the viral concentration method. To titrate the viral titer, each of viral pellet was resuspended with 300 microliters of media and stored in cold room for 4 hours. The virus was diluted 200 times and filled in 6 different volumes of the previously seeded HEK cell in 24 well plate as shown. This process was called HEK cell transduction. After 4 days of incubation, the GFP positive percentage was determined by flow cytometry and then calculate the viral titer in transduction unit per ml following this equation. Next, the virus containing shRNA was transferred to infect or transduce to OCI AMLT cell line. This process is called OCI AML3 transduction. So the amount of OCI AMLT and virus have to be calculated. The equation of multiplicity of infection or MOI was used as the following. The MOI is the integration event per each of target cell. For example, MOI of 5 means that the 5 of virus will integrate into 1 OCI AMLT cells. Mean of viral titer of each plasmid was calculated, and the MOI of 5 to 10 with approximately 3.5 million cells of OCI AML3 were used. The calculated viral stock was determined for each type of SESN1 that using to treat the OCI AML3. Both of transduced and untransduced OCI AML3 were cultured in 20% FBS in RPMI media. Finally, after 4 days of incubation, the OCI AML3 were washed twice with fresh media. Transduced cells were sorted by flow cytometry, and both of transduced and untransduced cells were extracted RNA, cDNA synthesis, measuring concentration, and perform quantitative real-time PCR with triplicate experiment per member pair. Target genes was SESN1, and internal reference genes were GAPDH and 18S. The second threshold or CT was generated and quantified the relative expression of SESN1 by delta-delta CT method. For the results, after isolated and measured the plasmid DNA concentration, the SESN1 antiviral production was performed by transfection of HEK290 T cells with corresponding plasmid. And GFP positivity were also confirmed in all plasmid as this picture. To determine lentiviral titer, the virus containing media at 48 hours of transfection was collected and concentrated by ultracentrifugation. HEK cells were seeded and transduced with 6 different volumes of diluted virus and determined transduction efficiency by percentage of GFP. This table shows percentage of GFP positive in each of SESN1 type at different volume of lentivirus. Subsequently, we used percentage of GFP to calculate the viral titers by using this equation, and the mean viral titer of SH1 and SH2 targeted SESN1 were 64.2 million and 55.9 million transaction units per ml, while mean viral titer of Scamber was 20.5 million transaction units per ml. To demonstrate the silencing effect of SESN1 targeted SESN1, OCI AML3 were transduced by lentivirus using the equation of MOI to calculate the viral stock. Due to limited amount of viral stock, we have to fix the MOI at 5 that resulted in using different number of OCI AML3 cells as shown in the table. And we also used 3 million cells of OCI AML3 without using virus, or we call untransduced cell to be another negative control. Lastly, after 4 days of transduction, all transduced cells were sorted and selected by GFP positivity. Both of transduced and untransduced OCI AML3 cells were performed SESN1 expression by real-time PCR. By comparing to untransduced cells, SH1 and SH2 targeted SESN1 significantly decreased expression of SESN1 when using both of internal reference genes. When compared to Scamber, SESN1 also shows significant reduction in SESN1 expression for both of SH and T type. This experiment confirmed the efficacy of lentiviral SESN1 to silence the SESN1 expression. This study aimed to evaluate the efficacy of SESN1 in silencing SESN1 in the transcription 69 AML setting. The technique of transfection and transduction were performed. However, there were several limitations and future perspectives to be discussed. Gene silencing technique in this study used the RNA interference machinery. Both of small interfering RNA or siRNA and small hairpin RNA or shRNA can directly be used to transfect into target cell, but this process results in transient silencing. Likewise in this study, using the DNA plasmid that encodes shRNA and packaging into the virus that proceeding to infect or transduce to target cell will result to stable indication and silence gene expression. SESN1 will be processed in the nucleus and being exported to bind the RNA-induced silencing complex in cytoplasm that subsequently degrade the certain mRNA and knocking down expression of the gene. SESN1 encodes cysteine protein that the production is triggered by p53 level. This protein also responds to oxidative stress and DNA damage by reducing the reactive oxygen species. Moreover, cysteine also related to increase the level of AMP-activated protein kinase and autophagy that subsequently mediate to cell growth suppression. Therefore, the regulating of physiological response of SESN1 in the transcription 69 AML is an exciting strategy to understand the oncogenic mechanism that driven by NUP214 and may provide a rationale for a targeted therapy in this patient. However, there were several limitations and tentative future perspectives. Firstly, transfection efficiency under fluorescent microscope in this study was qualitative measurement by visual inspection. The numerical assessment by using image analyzing software may improve the quality of further experiments. Next, the transfection method in this study was chemically treated by calcium chloride. Other alternative methods such as electroporation or transferring of non-viral genes may result in different outcomes. Moreover, the viral concentration method used in this study was ultracentrifugation that may result in instrumental barrier in the lab with limited resources. Alternative method, for example, the sucrose gradient centrifugation with a low speed of centrifuge, has been shown as an effective viral concentration method. Lastly, to enhance transduction efficiency in further experiment, the transduction enhancer and other techniques should be considered. The positive charge cationic polymer like polybene with using of transduction inhibitor removal kits can enhance the cell-to-virus contact. Other techniques like reverse transduction by coating virus on the vessel surface before seeding the target cell, and the spinoculation by adding centrifugation in transduction process may increase the transduction efficiency in the future. In conclusion, this study shows silencing SESN1 by using SSRNA, and the successful of decreased SESN1 expression in OCI-ML3 was present. This study also served to be the model experiment for further use in ML cell line with transcription 69 or FKH1, the primary ML sample, as well as patient-derived xenograft mouse model. The apoptosis assay, cell second, and differentiation assay after knocking down SESN1 should be performed to review the related oncogenic mechanism that SESN1 might be targetable in ML patients with transcription 69 in the future. Thank you for your attention.